What is carbohydrate metabolism? Carbohydrate metabolism denotes the various biochemical processes that are responsible for the formation, breakdown, and interconversions of carbohydrate in living organisms. Glucose, which is simple sugar monosaccharide, is a central molecule in the carbohydrate metabolism. Energy obtained from metabolism in the oxidation of glucose is usually stored within cells in the form of ATP. Organisms capable of aerobic respiration metabolize glucose and oxygen to release energy with carbon dioxide and water as byproducts. Carbohydrates are the main part of our food that present in the form of polysaccharide, disaccharide, and monosaccharide. After digestion, these all forms are converted to the sugar that may be present in the form of glucose, fructose, and galactose. There are 5 major pathways of carbohydrate metabolism. The first one is glycogenesis, which is the formation of glycogen from glucose in the liver. Second one is glycolysis. Glycolysis is the oxidation of glucose to pyruvate and lactate that take place in a cytosol. Third one is glycogenolysis, which is the breakdown of glycogen to glucose. Fourth one is gluconeogenesis, which is the synthesis of glucose from non-carbohydrate precursors such as amino acid and glycerol. And the last one is Krebs cycle. Krebs cycle is the oxidation of acetyl-CoA to carbon dioxide. It is the final common oxidative pathway for carbohydrates, fats, amino acid through acetyl-CoA. These are the steps of glycogenesis. First of all, glucose in the presence of the enzyme glucokinase or hexokinase gets converted into glucose 6-phosphate. Here, one molecule of ATP gets converted into ADP, thus energy is required. If glucose is present in the liver, then the process is catalyzed by glucokinase. But if glucose is present in the muscle, then the process is catalyzed by hexokinase. Now, glucose 6-phosphate gets converted into glucose 1-phosphate, and this conversion is catalyzed by phosphoglucomutase. Now, UDP glucose is formed, which is uridine diphosphate glucose from glucose 1-phosphate, and UDP in the presence of the enzyme UDP glucose phosphorylase. To initiate the process of glycogenesis, primer is required. UDP glucose is important for addition into preformed glycogen polymer. The preformed glycogen polymer will be denoted as N, and now it will be attached with UDP glucose that catalyzed by glycogenine. Hence, it will form glycogen and it will increase the change by 1. This process is catalyzed by glycogen synthase, which is the rate-limiting enzyme.
Next is glycolysis. Glycolysis is the first step in the breakdown of glucose to extract energy for cellular metabolism. Specifically, this process turning one molecule of glucose into 2 molecules of pyruvate. Glycolysis takes place in the cytosol of a cell and can be broken down into 2 main phases. The phase 1 is energy requiring phase. In this phase, glucose gets rearranged and 2 phosphate groups are attached to it. The phosphate groups makes a bona fide sugar called fructose 1,6-biphosphate. Fructose 1,6-biphosphate splits to form 2 3-carbon sugars called DHAP and glyceraldehyde 3-phosphate. Because the phosphates used in these steps come from ATP, 2 ATP molecules get used up. Only one, which is glyceraldehyde, can directly continue to the next step of glycolysis, while DHAP will be converted into glyceraldehyde diphosphate, so both finish the pathway in the end. Phase 2 is energy releasing phase. Each 3-carbon sugar is converted into another 3-carbon molecule called pyruvate through a series of reactions. In these reactions, 2 ATP molecule and 1 NADH molecule are made. Because this phase takes place twice, which is 1 for each of the 2 3-carbon sugars, then it makes 4 ATP and 2 NADH. Overall, glycolysis converts 1,6 carbon molecule of glucose into 2,3 carbon molecules of pyruvate. The net products of this process are 2 molecules of ATP and 2 molecules of NADH. Glycogenolysis. It is the process by which glycogen, the primary carbohydrate stored in liver and muscle cells, break down into glucose to provide immediate energy or to maintain blood glucose levels during fasting. This process is stimulated by the hormones glucagon and epinephrine. For example, when blood glucose level falls, there is an increase of glucagon secretion from pancreas and it will travel to the liver to stimulate glycogenolysis. Here is the process of glycogenolysis. In the liver, glycogen is converted to glucose 1-phosphate by the enzyme glycogen phosphorylase. Then, glucose 1-phosphate is converted to glucose 6-phosphate by the enzyme phosphogluconeogenesis. The liver tissue also houses the enzyme glucose 6-phosphatase, which converts glucose 6-phosphate into free glucose that is secreted into blood, thereby restoring blood glucose levels to the normal.
Fasting state is a condition where the body is not receiving any food. So, the body has come up with two different ways in regulating blood glucose level. One is by breaking down the glycogen stored in the body into glucose. Liver is one of the most important organs in carbohydrate metabolism because most of the glycogen is stored in the liver. However, the glycogen stores only last 10 to 18 hours in the body before being used up. So, the body has come up with a second way, which is gluconeogenesis. The word gluco means glucose, neo means new, and genesis means production. In normal circumstances, carbohydrates are broken down in human body by various enzymes into glucose. In special circumstances, pyruvate, fat, and amino acids are converted into glucose. So, gluconeogenesis can be defined as synthesis of glucose from non-carbohydrate sources. Gluconeogenesis is one of several main mechanisms used by humans and many other animals to maintain blood glucose level, avoiding hypoglycemia. Gluconeogenesis is a ubiquitous process present in animals, plants, bacteria, fungi, and other microorganisms. Sites of gluconeogenesis: liver, kidneys, and intestines. Why gluconeogenesis is important? First, to prevent low blood glucose level. Second, brain completely relies and depends on glucose because glucose is primary source of fuel for brain. Gluconeogenesis is almost the exact reverse pathway of glycolysis. Glycolysis breaks down glucose into pyruvate, while gluconeogenesis produces glucose from pyruvate. It starts with pyruvate and it goes funnel back the opposite direction through all of the glycolysis reactions to produce glucose. However, almost doesn't mean totally same. When comparing the two reactions, the different things that occur in gluconeogenesis can be seen in three phases as shown here. The body has come up with a different reaction pathway for the three irreversible steps. TCA cycle is tricarboxylic acid cycle, is also known as the citric acid cycle, and the Krebs cycle is a series of chemical reactions used by all aerobic organisms to generate energy through the oxidation of acetate derived from carbohydrates, fats, and protein into carbon dioxide. The name of this metabolic pathway, such as TCA cycle, is derived from three carboxyl groups on its first two intermediates, while citric acid cycle refers to the first molecule formed during the cycle reaction, which is citrate or citric acid, and the Krebs cycle is after its discoverer Hans Krebs. The cycle consumes acetate in the form of acetyl-CoA and water, reduces NADH to NADH and produces carbon dioxide. The NADH generated by the TCA cycle is fed into the oxidative phosphorylation pathway. The net result of this two closely linked pathway is the oxidation of nutrients to produce usable energy in the form of ATP. Acetyl-CoA is produced through the catabolism of sugars, fats, and proteins. One of the primary sources of acetyl-CoA is sugars that are broken down by glycolysis. Glycolysis produces pyruvate that is decarboxylated by the enzyme pyruvate dehydrogenase, producing acetyl-CoA. The TCA cycle takes place over 8 different steps. single turn of the cycle, 2 carbons and 2 molecules of carbon dioxide are released, 3 NADH and 1 FADH2 are generated, and 1 molecule of ATP is produced. So the product for each glucose can be obtained by multiplying the product of a single turn of the cycle with 2.